Hello, hello, hello. Okay, then I guess we'll begin. Hi, everybody. My name is Oscar Garcia, and first of all, I want to thank you, every one of you, for being here. And I want to thank Kabir for helping me and allowing me to be here with you today. And well, let's just begin. Today, we're going to talk about the safety and efficacy of an RID26 and RAD5 vector-based heterologous prime boost COVID-19 vaccine. This was a randomized controlled phase 3 trial in Russia. Let's talk a little bit about it. Well, first, we've been through a tough situation. Hello? Somebody want to say something? I guess not. As I was saying, we are now going through a tough situation because of the pandemic we're going through. Since we are facing this new virus and this pandemic of COVID-19, at this date has been more than 120 million cases around the world and more than 2.7 million deaths worldwide. This has led to the World Health Organization to take response and make some vaccines for developing safe and to focus and develop prevention for this virus, COVID-19 virus. At this day, we have few provisionally licensed vaccines. It's been known that at this date we have already 13 vaccines at phase 3. We have 64 candidates in clinical assessment and 173 in preclinical analysis. We have different types of vaccines. Between them, we have the ones that are based on vector, like the vaccine we're going to talk about in this trial, or about this trial, and as well as known as Sputnik 5 or the gun COVID vaccine. We have some mRNA based vaccines like Moderna and Pfizer. We have inactivated vaccines like Sinovac, and we have some adjuvant recombinant protein nanoparticles like the Novavax vaccine. Well, it's well known at this date that adenoviral vector vaccines, like the one that we're going to talk about today, they are good for inducing both cellular and humoral immunity. And adenoviral vector vaccines have been studied extensively, and there's at this date a few therapeutic drugs that are in use at the clinical practice. Adenoviral delivered antigens are known to induce both the cellular and humoral immunity, as I was saying. It's this reason that after a single, what? Somebody say something? Okay. We can produce both of these immunities by giving just one or a single dose of a vaccine that is an adenoviral vector vaccine. This allows their use as an emergency prophylaxis tool in a pandemic, as it goes in this time we're facing. What is best, or is better, sorry, is that if you use two immunizations, you're getting a durable and long-lasting immune response. That's why they are good candidates for long-term protection of the people at high risks, because they stimulate rapid onset of protective immunity. There are also some questionable things about the adenoviral vector vaccines, because they induce some immune responses against the vector components. This can make that the efficacy of the vaccine gets a little bit less effective. But if you use this type of vaccines on an heterologous vaccination with two different vectors, as it goes in this type of vaccine, the Sputnik 5, this allows minimization of this problem. And even better, if you use this type of vaccine, it gets even more effective as a powerful and long-lasting immune response. Well, now, what is the GAM-COVID vaccine, or the Sputnik 5 vaccine? This vaccine, as I was talking earlier, it's composed of two recombinant replication deficient adenoviruses, which are the replication deficient adenovirus type 5, or RID5, and the RID26. These adenoviruses, sorry, carry on inside them the gene for the SARS-CoV-2 full-length glycoprotein S, that it's going to be the component that's going to make in the body of the person that's getting vaccinated the immunity response, and then the immunity for the COVID-19. Once you get the SARS-CoV-2 full-length glycoprotein S inside those RID, you get as a combination the vaccine, or the Sputnik 5 vaccine. There is two doses. They apply there. It applies as a two doses in a interval of 21 days between them, and each one carries on one of the adenoviruses. In the first vaccination, you get one of the RID, and the other vaccination you get the other. Well, now talking a little bit more about the Sputnik 5 vaccine. It finished the phase 1 and 2 trials on August 21, the past year, and in those trials, it seemed like it was well tolerated and highly immunogenic in healthy participants. Because of this, it was provisionally approved in Russia according to their national legislation, and then was started to apply in high-risk groups with enhanced pharmacovigilance. This was a point that around the world was a little bit untrusting. Like it was really criticated because it was really fast that this vaccine started to get into the people there in Russia. Well, now at this trial, we're going to review a phase 3 randomized trial, so we can see if this vaccine really is really helpful or not, and if it is really safe or not. Well, let's first let me first say that this was a study and that it was randomized, double-blind, placebo-controlled, and it was a multi-center phase 3 trial in Moscow, Russia, and it was done at 25 hospitals there in Moscow, where the Health Organization of Russia was in charge of all of this study. It was made to assess the efficacy, immunogenicity, and safety of this vaccine. Well, how the participants could get into this trial? Well, there was some eligibility criteria, and in this criteria we get the participants should be at least 18 years old or older. They have to be negative to HIV, hepatitis B, C, and syphilis. They had to be negative and IgM and IgG and antibodies for SARS-CoV-2 and PCR tests. They needed to have no history of COVID-19 or of history of being close to somebody with COVID-19 in the past 14 days before. They had to consent to use effective contraceptives methods in case of the woman, They had to not be pregnant, to have a negative urine pregnancy test. They have to not use drug or alcohol, have no history of vaccine-induced reactions, and to not have acute infectious or respiratory disease in the 14 days before enrollment. The exclusion criteria it's a little bit more extensive, as some of the most important is that they they haven't got any vaccination, or they have any vaccination in the 30 days before enrollment. That was an exclusion criteria. That they don't have any acute coronary syndrome or stroke in the year before enrollment, any allergy or hypersensitivity to the drug or components of the vaccine, and that they don't uh, that they if they uh, had donated blood in the 2 months before enrollment, they couldn't get into the study. And as it was said before, if they have an active form of a disease like HIV or hepatitis, they couldn't get into the study. Any deficiency on proteins or anorexia, they they too can't get into the study. Well, let's pass this. Sorry. Well, how was the randomization and masking of this trial? The participants got into 5 age strata, and they they make 5 groups of people ranging from 18 to 30 years old, 31 to 40 years, 41 to 50 years, 51 to 60 years, and more than 60 years. Then the participants got to 2 groups in a ratio of 3:1 to the vaccine or the placebo group. The drug, the vaccine, and the placebo were outwardly indistinguishable. The packaging, the label, the content, the color, everything was the same. And in participants, investigators, and all the staff that was in the study were masked to the group segment, and they couldn't know whoever gets the vaccine or whoever gets the placebo. Well, oh, sorry. At the start, 35,963 participants screened to this study, and 13,986 were not eligible or not yet randomized, and the 21,977 got randomized or got into the study. From those, 16,501 got assigned to the vaccine group and received the first dose of the vaccine. 5,476 assigned to placebo group and received the first dose of the placebo. From those that got the vaccine, 74 got excluded because of a protocol violation. And at the end of that, 16,427 got included in the serious adverse events analysis that we're going to talk about it more later. And in the placebo group, 5,435 35 got included in the serious adverse events. From those, 14,964 received the second dose of the vaccine and were included in the primary outcome analysis that we're going to talk about it later too. And 4,902 received the second dose of the placebo and were included in the primary outcome analysis too. At the end, in the immunogenicity analysis, I got 342 got included into the receptor binding domain specific IgG analysis that we're going to talk later too. 72 included in the neutralizing antibodies analysis, and 44 included in the IFN gamma analysis. From the vaccine group, and from the placebo group, 114 got included in the receptor binding and domain specific IgG analysis, and 28 included in the neutralizing antibodies analysis, and 14 got included in the IFN gamma. We're going to talk more about all of this later. Well, what was the procedures for this trial? All participants screened for a visit at the beginning, and the intended duration of the participation in this trial was 180 days after the first dose, and they will have one visit screening. Somebody want to say something? They will have one screening visit and 5 on-site visits to a clinical site over the course of the trial. The vaccine, as I was telling you, was somebody want to say something? Was compounded by 2 vector components, the RID26S and the RID5S, as I told you earlier. Well, every dose of the vaccine was 10 to the 11th potential viral has this 10 to the 11 viral particles per dose for each of these recombinant adenoviruses, carrying on the gene of the SARS-CoV-2 glycoprotein S. Every dose was 0.5 milliliters, and it was an intramuscular injection that was put into the deltoid muscle. The placebo consisted of the vaccine buffer without the recombinant adenoviruses, and it has those were used in the liquid form. This means that they were frozen at minus 18 degrees Celsius, and we're gonna talk a little bit more about this later too. The vaccines were administered, as I said, intramuscularly within a 21-day interval between both doses, and the subsequent observation visits were planned for the days 28, 42, and 180. The blood sampling was done on the first day of vaccination immediately before studying drug administration. The antigen specific IgG analysis were planned from up to 9,520 trial participants. This was analyzed on the day of the first vaccination and on the day 42 after the vaccination. These procedures were ascertained by ELISA, and it measures the specific IgG to the RBD of glycoprotein S, the receptor binding domain of the glycoprotein S. The seroconversion rate was calculated as a fourfold increase in the titer at 42 days compared with the day before the first vaccination too. They too analyzed the cell-mediated immune response, and this was measured by quantification of the IFN gamma secretion. We'll see later their results. And they made this, or they quantified the secretion of the IFN by stimulating the immune response of the participants and giving them a restimulation with an antigen of SARS-CoV-2 glycoprotein, and they will measure it and on peripheral blood cell culture of their mononuclear cells. Well, what were the outcomes of this trial, of this study? The primary outcome was the proportion of participants with COVID-19 that was confirmed by PCR from day 21 after receiving the first dose. This means they will measure how much participants get COVID-19 after 21 days before they get the first vaccine, and that means that they started counting after the second dose. At the 21 day, the participants were gonna receive the second dose, and after they get the second dose, they will be measured how much people, or how much persons on the participants, on the study get the COVID-19. The secondary outcomes were to measure the severity of COVID-19, changes in antibody against glycoprotein S, as I mentioned earlier, the proportion of participants with antibodies against N protein of the SARS-CoV-2, the changes in their neutralizing antibody titers, their changes in antigen specific cellular immunity level. We're gonna see all of this later. And the incidence and severity of adverse events. Sorry. Well, and while making this trial, 78 COVID-19 cases were found on participants after receiving the second dose. The frequency in the placebo group were expected to be 20 per 1,000 or 2.0%, like in the general population. And this study aimed at the proportion will be at least a third lower in the intervention group. And they expected a value of the effect at about 0.5 or 50% at least. Well, and the primary endpoint was calculated using the following formula. On the vaccine efficacy, on percentage was equal to 1 minus the odds ratio plus 100, where the odds ratio is as follows, we all know. And the mm, the oh, sorry, equation, no, the formula, sorry. Where in this formula in this formula, A is the number of vaccinated participants with COVID-19. B is the number of vaccinated participants without COVID-19. C is the number of unvaccinated participants with COVID-19, and D is the number of unvaccinated participants with COVID-19. Well, using this, we get the results of this trial, and it went from between September 7 and November 24 of 2020, the study. On here, 21,977 adults were eligible and randomly assigned to receive the placebo or the vaccine. 74 participants from the vaccine group and 41 from the placebo group were excluded, as I told you when we saw the image earlier. And the preliminary analysis included 16,427 participants in the vaccine group and 5,435 into the placebo group. 14,964 and vaccine and 4,902 in placebo had received 2 doses at the time of that database lock that was on November 24, 2020. On the results, well, the vaccine efficacy at any time after dose 1 was 73.1%. This means that if you get vaccinated today, you, in this right moment, you get at least 73.1% of protection. But this has a little trick there. We're going to talk about it a little bit later. What it is certain is that 21 days after the first dose of the vaccine, in this case, the day you're going to get the second dose of the vaccine, you're getting a vaccine efficacy of 91.6%. That's for sure. And they saw in this study that 16 cases were presented in the vaccine group out of 14,964 participants after 21 days of the first dose. That means that 0.1% of the population that got vaccinated got COVID-19. In the other hand, in the placebo group, they got 62 cases out of 4,902 participants. That was 1.3% of the population. And something that is remarkable is that this vaccine, 21 days after the first dose, once you get the second dose, it got an efficacy against moderate or severe COVID-19 of about 100%. So if you get both doses of this vaccine, the GamCap or the Sputnik 5 vaccine, you're getting a 100% chance of not getting a moderate or severe disease from COVID-19. We'll see it later too. Well, as I was saying, the aim of this study was to see how much occurrence of COVID-19 were on all of these participants from 21 days after the first dose. And overall, they got what I told you, that this efficacy vaccine was 91.6%. Well, we all know that this disease tends to be more severe or kill more people that has older age. So if we see the efficacy and the group of more than 60 years, the vaccine has a almost the same efficacy. What was remarkable, as I said, was that the moderate or severe disease was null, or it doesn't present it in any of the participants that got vaccinated, while in the placebo group they got some cases of the severe disease. This means that this vaccine can protect at by 100% on moderate or severe disease. As I was telling you earlier, the efficacy of this vaccine before 21 days or after the first dose, it's lower. We'll talk more about it a little bit later, but at any time after the first dose, it got an efficacy of 73.1%, and from 14 days after dose one, they got an 87.6%. So we'll talk a little bit more about it, as I said, but it got proof that at any time from 14 days after the first dose, you get a good amount of protection with this vaccine. Well, the results on the analysis of humoral immune response, the samples were from